Everybody hear me? I'm good. Outstanding. Well, welcome everybody to the immunotherapy session. I'm co-chairing this session with Ari Hakimi. And there is a little change in the speakers. The first speaker, unfortunately, could not attend. And Ari was so kind to actually jump in. Theme-wise, it's going to be very interesting, I think. There will be some new thinking in how we look at T cells and identify some promising T cell populations for therapeutic purposes. And you target potentially to induce the immune response. And Ari will be so kind to actually touch on the topic of myeloid cell population and the influence on immunotherapy.
Thank you. And I'm very excited to co-chair this session and talk about some exciting science. May I have slides, please?
You don't see any slides.
Yeah.
Present maybe?
Can you go back? Yeah. Keep going. Yep. One more. Thank you. Okay, so it's my pleasure today. This was the last minute, so I apologize if there's any confusion in any of the slides. I'm happy to address them afterward. But so I'm going to talk a little bit about our recent work that was just published on targeting the myeloid response in clear cell renal cell carcinoma. This is my disclosure, nothing to do with the talk today. So I think we all have been appreciative of the landscape of how we are understanding the immune response in clear cell, in particular kidney cancer, has expanded quite dramatically over the last several years. In particular, single cell sequencing efforts by large institutions such as Tony Chouari's group and others from the Broad, Elliot Van Allen, as well as Chuck Drake, and then our own work at MSK has really kind of given us a broader sense of what populations are present both early and metastatic kidney cancer, which I think opens up opportunities for therapeutic development. And in our own work, we had looked at patients that were both untreated and post-Ipi-Nivo and kind of described more of a broad sense of the dominant populations. And I think most of us were aware that clear cell renal cell carcinoma is very T cell enriched, and it is in fact probably 50% or more of the immune populations. But clearly there are other populations that are present and are probably serving roles that could potentially lead to therapeutic strategies. And in particular, our work identified 4 or 5 different major macrophage populations amongst many other populations. One thing I should point out, in most of the single cell studies that were done, there was a step where the tissue was frozen at some point, and that really depletes things like neutrophils, which probably serve a very important role as well. And I will not be talking about that, but I think it's something that we're increasingly aware of and have adjusted our own single cell strategies to account for. And in that study, we identified the different immune populations, and I think something that's emerged from other studies have kind of pointed to the role of the myeloid populations, and that's a broad term. Often these are characterized by transcriptional signatures, which is really stating more of a state and may incorporate many different immune populations. But work from the Genentech and led by Dave McDermott and others had clearly demonstrated the impact of myeloid enrichment scores in terms of stratifying patients by response to single agent and combination immunotherapies. And in fact, in the recent work that was presented the recent work that was published and referenced by many at this meeting, there's a sense of dominant clusters that represent different mutational enrichments and their association with specific microenvironmental states. And in point of fact, the stromal proliferative cluster that Brian discussed and others have discussed is enriched for p53 mutations, which are relatively rare in localized disease. But if you look at a lot of the metastatic sequencing cohorts in some of the larger pivotal phase 3 trials, it represents about 10 to 15% of mutations in advanced kidney cancer. So it is clearly a population that is a group of tumors that's very relevant. So we had started to see this a couple years ago in work that we had done in focusing on VEGF TKI space, and we had looked at the COMPARES trial, which had compared sunitinib and pazopanib, and demonstrated that in tumors that didn't respond to pazopanib or sunitinib, for that matter, they were enriched really for macrophages. So the dominant feature of these tumors that were not responding, besides having lower angiogenesis, was really an enrichment of macrophage population. So suggesting that there certainly could be a biomarker for a lack of response to VEGF-TKI-based therapies. So in this recent study that we published, we actually took this now to a different question, and this was really looking at what is the role of the immune microenvironment with respect to tumor recurrence. And I think a lot of this dovetails nicely with the previous session that talked a lot about adjuvant therapy. And I want to point out that the questions I think are a little different. So many of us are thinking about what are the populations that are associated with therapeutic response. But when you're looking at an adjuvant cohort, you're really looking at what are the populations associated with development of metastatic disease. It's a little different question. I think a lot of us have focused the same therapeutic strategies on how we treat metastatic disease, but maybe the concept of micrometastatic disease or recurrent disease is a little bit different. So we looked at the PROTECT trial. This was in collaboration with Novartis and led by Dr. Motzer, myself, and Dr. Voss. And we looked at the placebo arm. So we focused on untreated patients, and we had microarray actually, and IHC and clinical pathologic data. And I'll talk mostly about the microarray data for the purposes of time here. And we looked at what are determinants of recurrence in this population. So again, these are all high-risk patients. They're all fit the bill, and we're looking at the placebo arm here. And so the first thing we did, and this work was led by a urology fellow, Phil Rappold, and a very, very talented postdoctoral student, Lin Fong, who's been in my lab for several years. And we looked at, again, what were the pathways that were enriched in patients that were developing recurrence. And some of the top hits, of course, were things that we would expect, like EMT and mTOR, for example. But what we noticed was there was a lot of immune-related populations that were really enriched. And this kind of centered around IL-6 and a lot of myeloid signaling pathways, which was really intriguing to us. So we looked at our single cell data that we had generated from our previous effort, and we identified sort of what are these populations really enriched for, and we identified several signatures that had been previously published, including this myeloid signature that Genentech had developed, as well as an adenosine signature, which we'll come back to a little bit, that was developed with Corvus, which is a company that's been developing therapies to target myeloid populations in the form of adenosine inhibitors. And so we looked at whether these were kind of capturing these macrophage-related populations that we had identified in the primary tumors that we had done single cell on. And sure enough, we saw a lot of enrichment in this. So we developed sort of our own inflammatory score where we combined several of these populations. We vetted them against kind of TCGA data sets to try to really understand if this is uniquely enriched in clear cell renal cell carcinoma, which we did find. And we kind of developed a score that would be helpful to generate to see if this was predictive. And working together with our own data set, TCGA and Brandon Manley's, who's here from Moffitt, we kind of developed an inflammatory signature. And what you can see here is this myeloid signature, beautifully stratifies patients by recurrence. So these are all high-risk patients by pathologic features. But if you take a myeloid signature, you can stratify them into quartiles. It's pretty impressive. And this was reproduced in multiple data sets. So this inflammatory signature, myeloid signature, is really driving recurrence in these patients. And if you think about sort of our current therapeutic strategies, we're not necessarily targeting these populations with our current treatment strategies, and kind of opens up the concept of whether this would be a better approach to do that. And one thing that we noted is that, again, when we went back to the cancer cell data from Genentech, Dr. Motzer, we did confirm in many data sets that these tumors that were inflamed were also P53 enriched tumors. These were much more likely to have P53 mutations, which may potentially link some of the genotype to the immune phenotype in these tumors. So that was really important. But I think one of the things that we struggled with this time was how to model this, because we don't really have good immunocompetent models. So together with the Scott Lowe lab at MSK, we devised, and this was really work led by Lynn over many, many years of painstaking effort. We devised a strategy to develop immunocompetent systems in the Black 6 mice. And for this one, we utilized a strategy of genes that had been previously been successful in a GEM model led by Ian Frew from Germany, where he had deleted VHL, P53, and RB in mice and had developed a very nice clear cell model that was published in Nature Medicine a couple years ago. The problem with that model, which we have in lab and have been using for several years, is they develop multifocal tumors. They're very back-crossed, and they're very hard to develop any sort of lines from. So we utilized that strategy together with the MYC Sleeping Beauty transposon and developed basically an immunocompetent model that is syngeneic and can be easily transplantable. And importantly, this model metastasizes with quite vigorous intensity. And then, of course, we wanted to confirm that this model retained the features despite its very high-grade phenotype in terms of histology. But of course, we did show that it's HIF1 positive CA9 GLUT1. Our HIF2 characterization is ongoing because it's a trickier stain to do in mice, but it does appear to have HIF2 activity as well. And so this retained, despite its high-grade feature, all the models, all the hallmarks of clear cell renal cell carcinoma. And one of the interesting things is that we then performed RNA sequencing on this model and tried to see if it did recapitulate this kind of stromal proliferative model that was shown as one of the subgroups in the Motzer et al. Paper. And indeed, we found that the key features of that myeloid inflammatory proliferative subgroup are retained in our model. So this is really, I think, a very good human mouse model that represents a very aggressive subgroup of kidney cancer that's likely both metastatic and also probably resistant to some of the conventional therapies that we give. And because we had shown this was an adenosine high tumor type, adenosine high by transcriptome, and this is based on this beautiful work that came out of UCSF and Corvus, we in collaboration with them, utilized their agent, and were able to show that in this model that we developed, they could abrogate the development of metastasis, which were completely, by the way, PD-1 independent. So PD-1 did not slow down tumor growth or development of metastasis, but targeting a myeloid approach through an adenosine A2AR receptor inhibitor was a successful strategy in this model. So I think the takeaways here is that we know that clear cell renal cell carcinoma is heavily immune infiltrated. There are prevalent and dominant myeloid populations, many of which are associated with this p53 genotype. I think that the myeloid TME is very relevant probably and maybe driving a lot of the metastatic development that we see, which could hopefully help us inform potentially newer strategies for the adjuvant populations. And that targeting these populations might help us drive newer therapeutic strategies. And I think we're very excited to work with a lot of some companies now in sort of kind of defining subgroups that might best benefit from these strategies. And I just wanted to thank my group and, of course, all the funding sources that have helped support this effort, including the DoD. Any questions at the end, or would you question?
So thank you so much, Ari. We have some time for questions. Isaac.
Very nice talk. So, my voice is not enough.
You have a microphone?
The p53 linkage to the inflammatory myeloid signature you have, and if you want to talk about the 9p loss, because many groups, including our own, find that these aggressive tumors that you're calling myeloid are also driven, by 9p loss. Can you comment on that?
Yeah, so I don't think, I'm not an expert in terms of p53 pathways entirely, but I do believe that it's not necessarily a defined rationale, although others have shown p53 mutations linked to a higher myeloid inflammation, and I'm certainly, would be great to hear if anyone has a direct answer. So I don't know the answer, I just know that we've seen it on multiple data sets where we see this. In terms of the CDKN2A loss, which I think is, and B loss, which I think is the 9p, that certainly is something that you see in these proliferative, in fact, their cell cycle high, these are very aggressive tumors, and in fact associated with sarcomatoid development quite a bit. I didn't talk about that, but we've developed another model that also incorporates both BAP1 and CDKN2E in a syngeneic setting, and we clearly see that linking to sarcomatoid and also a higher myeloid state. So there may be multiple ways of reaching that. And again, the myeloid is a very generic term, so obviously it could certainly link to very specific macrophage, for example, subgroups within those different states. And I think it's an excellent point.
Great. Next question.
might have already answered my question. So my question was going to be, in terms of additional genetic combinations, did you go side-by-side comparison to ask, is this the best one?
Yeah, no, and I think every model has limitations. We chose this model for a variety of reasons, but also we had tried a lot of the other models over the course of 3 years, including this electroporation-based approach. I think when you take less aggressive, although certainly interesting subgroups such as PBR1-driven tumors, which are more angiogenic in general, they're harder to develop lines from. And I think Bill's shown that in his own data from attempting to do that with electroporation approaches also. It's a trickier group, so we have been successful with BAP1 and CDKN2A because, again, these are very aggressive phenotypes. Part of the problem is they don't look beautifully like clear cells, and I think we would all love to see that, but I think the challenge of those are a more differentiated state that may be harder to engineer, honestly.
Just a very quick follow-up. Have you ever tested existing therapeutic standards against the tumor?
Yes, so they're quite sensitive to some of the TKIs. We've used Cabo and Lenva, and they're quite sensitive. PD-1, they're fairly resistant to, and then they're quite sensitive to some of the myeloid inhibitors that we've been testing, but certainly interested in testing more. Hi, Jim.
Yeah, hi. So I just have a comment, and it's really a commendation to you. This is something that the field needed, the development of cell lines that are in a syngeneic background with the mutations in kidney cancer. Obviously, RP is not great, but this may be a first-generation cell line, and there are similar efforts at MD Anderson with Gianni, and of course with Bill Killing at the Harvard. So I think this is terrific. We are also trying at UT Southwestern, and I would encourage anybody who's been using the Renca to try to switch away from that model. I know Ari and Gianni are willing to share their cell lines, and probably the group at the Harvard as well.
Absolutely, we're working on master agreements for people. We'd love to share these lines.
Great. Any other questions over there?
Thank you, Ari. I think you have received lots of questions when you were publishing this work, but my question is that you know that we have tumor subpopulation, and these are basically coming from like single cell data. So, and we all know that because of the depth of sequence, it's very difficult to find like mutation behind that. So how could you, find like P53 is behind that subpopulation? So, how did you manage to connect sequencing of DNA to this subpopulation of cancers?
Yeah, I think it's a great question related to heterogeneity. One thing that we've looked at about how specific this finding is in unpublished work, but it's under revision right now, is if you take multi-regional sequencing, for example, where you have distinct evolutionary trajectories, for example, development of P53 is generally a later event. It's not a fundamental driver, which is, again, one of the limitations of our model, obviously. But, and then to see if, or CDKN2A loss, for example, and whether that's associated with distinct immune niches, for example, in the tumor. And indeed, we have found that if, for example, in CDKN2A loss, which occurs later, it is associated with a myeloid response in a particular region of the tumor, suggesting there is some direct relationship between that. But clearly, it's still bulk-related analysis.
Sounds good. One last question, maybe? Tony.
Do you think the signature for, I'll use both. Do you think the signature for adjuvant could be the same use for metastatic? We don't want to go back and forth. You saw some similarities.
Yeah, no, I think so. I mean, I know Corvus has a cooperative trial that they're currently doing with MD Anderson and others, and I think I would love to start seeing some of this approaches being utilized. There's a lot of myeloid inhibitors that are being kind of in development. I think we'll talk at another meeting about the limitations of CSF1, which is what had been a total dud. But if you look at sort of the population that it's targeting, we know it targets a broad range of myeloid populations, probably some really beneficial ones and some bad ones. And so there's a lot of opportunity for precision here.
Outstanding. So I would say we move on to the next speaker. The next speaker is Hudi Warren from Washington. And It's going to be about identifying interesting T cell populations.
Everybody hear me? Yes. Good, thanks. I just wanted to thank the organizers for the opportunity to be here today. I have learned an enormous amount over the past 36 hours, and it's really just incredibly exciting. I have to disclose no conflicts of interest, but I want to tell you all that I am not a kidney cancer doc, and but I'm very fortunate, and I get to work with a lot of very good kidney cancer docs. So keep that in mind, and I'll mention, of course, some of them along the way. I went to Seattle some long time ago to really learn to dissect the graft versus leukemia effect associated with allogeneic hematopoietic cell transplantation at the cellular and molecular level. And that the cellular dissection of anti-tumor T cell reactions has been the focus of our lab over the intervening decades. And I really feel that if we can tease apart these anti-tumor T cell reactions at sufficiently resolute or a degree of resolution, we can define and really develop more effective immunotherapy for kidney cancer and a lot of other cancers. And so T cells recognize a variety of ligands, as we'll discuss in a second. They do so with their antigen receptors, and their antigen receptors are very convenient because they provide T cells with two unique barcodes, and those molecular barcodes allow us to track and enumerate T cells across space and time with exquisite precision. But not only do those nucleotide sequences in the variable regions of the antigen receptor allow us to track T cells, count them, etc., but they determine, of course, the antigenic specificity of T cells. And now with the advent of single-cell RNA-seq and single-cell proteomics, we can actually associate a T cell receptor antigen sequence with an antigenic specificity and a function, and really draw powerful conclusions about what drives anti-tumor T cell reactions. So I said T cells recognize a variety of things, and in contrast to what you might have learned in graduate school or medical school, not all T cells recognize peptide presented by MHC molecules. And there's a subset of T cells that recognize glycolipids presented by CD1B or CD1D. Those are important in microbacterial responses. And as you may or may not hear from Dr. Sewell, there's a very important subset of T cells that recognizes vitamin B and riboflavin metabolic intermediates presented by a non-polymorphic MHC1-like molecule called MR1. And in this slide, I've actually just given a shout out to Dr. Sewell and a paper that his group published a couple of years ago, because actually it's the inspiration for the DoD KCRP project that I'll tell you at the end of this. So my attention and interest in RCC was piqued by the publication of this ancient paper back in September of 2020. A group from NHLBI described very durable regression of metastatic kidney cancer in individuals who had undergone non-myeloablative allogeneic hematopoietic cell transplantation. So based on that observation, a group at the Hutch led by Brenda Sandmeyer started a comparable trial. And we started getting involved in the correlative science in those patients, trying to figure out what was going on and why. One thing was very clear from all the studies, many such groups undertook an effort, is that responses were clearly associated with the development of graft versus host disease. I want to tell you about really two clinical vignettes because they illustrate, I think, what we can learn about immunotherapy by doing intensive study of spectacular responders. And so this is a patient with metastatic clear cell who underwent an allogeneic hematopoietic cell transplant at the Hutch 22 years ago and had a very interesting clinical course. Suffice it to say that she did not respond until well after a year post-transplant, 3 donor lymphocyte infusions and alpha interferon, but she achieved essentially a complete response that proved durable, and unfortunately she passed away due to an infection, but in CR. several years after her transplant. And most of this work was done sorting out the T-cell responses in this patient by a fellow in my lab who's now a bona fide kidney cancer doc, Scott Cody. So what Scott found out is that most of the donor anti-recipient immune response in this patient was an alloreactive response, as you might imagine. Allelic polymorphism in genes across the genome really are quite immunogenic, and in an MHC-matched transplant really can serve as targets for T cells that have very potent anti-leukemic and anti-tumor activity. That occurred in this patient, and I've provided two examples here of T cells that Scott teased apart in his studies. One of them is a conventional gene, a Rho GATPase associated protein, but I wanted to point out that the other is actually a peptide presented encoded in a cryptic open reading frame of a gene that has absolutely no known protein product, and that's actually seen time and time again in tumor immunology, that peptides presented on the surface of cells are really not part of the curated, annotated, well-recognized proteome, but they're very good targets for immunotherapy. From this patient, we also really generated a lot of recipient-specific tumor-reactive T-cell clones whose antigenic specificity remains undefined, and that's where Dr. Sewell's work will be relevant, hopefully, because we've adopted an approach that developed in HIP's lab to identify what the targets of those T cells are. Many of them display MHC unrestricted T cell reactivity. So unfortunately, the toxicity of transplant for renal cell carcinoma proved prohibitive, and the procedure was abandoned. Fast forward to 2012, when this paper on the left appeared. And in the intervening 10 years, immuno-oncology and checkpoint inhibition, the inhibition of the PD-1 axis in particular, has become really a pillar of treatment for advanced renal cancer. Suffice it to say that even in October of 22, we really don't understand with sufficient resolution the T-cell responses that underlie response to immune checkpoint inhibition in metastatic kidney cancer or other malignancies for that matter. And I think there's really an urgent need to understand what's going at the molecular level, what's the breadth, the depth, the functional properties, and the antigenic specificity of those responses. So the second patient I want to tell you about is actually a patient with metastatic non-small cell lung cancer that was treated at Memorial Sloan Kettering by a colleague by several colleagues, and she was actually a patient of a colleague of mine from the University of Michigan, Nitha Ramnath. Suffice it to say, she had a very advanced multiply relapsed non-small cell lung cancer, and in July of 2013 developed metastases involving the buttocks and vaginal vault, and she got some palliative XRT in August of 2013. She enrolled on a clinical trial of atezolizumab, known as MPDL 3280A at that time, and achieved a durable response that she has maintained mostly maintained over 10 years since then, almost 10 years. And she developed some immune-related side effects, but really has had a remarkable quality of life. She is very happy to share her name. She's Judith Gray, and she lives in Ann Arbor, Washington. Her face and story are on the cover, on the website of the Cancer Research Institute. But we've studied this patient with extraordinary detail, thanks to Nithya and the availability of peripheral blood mononuclear cells and tumor specimens from this individual patient. And what's very clear that there was an oligoclonal massive expansion of T cells in her blood immediately after atezolizumab and radiation. And that has persisted for 10 years. And it's very likely that these T cells have something to do with the durable tumor regression seen in her. One of the interesting things about this particular response, there was one clone in particular that for most of the 10 years was 10 or 15% of her peripheral blood T cell repertoire. That clone had CD8 T cell effector profile. Very interesting antigen receptor characterized by very long alpha chain and beta chain CDR3 loops that I've indicated here. We haven't crystallized that T cell receptor, but thanks to AlphaFold, the the scientific breakthrough of 2021, we've actually predicted the structure, and the structure actually predicts something that looks unlike any T-cell receptor we've had before. Extremely long CDR3 loops with unlikely any role played by the CDR1s and 2s. And so although we think we know what T-cell antigen receptors see, I think that this particular T-cell receptor actually recognizes something completely different and not on this list. And I actually think it's a cell surface molecule that has nothing to do with class 1 molecules or MR1 or CD1B or CD1D. And so is there precedent for that? There really is, actually. There's Jim Yang from the surgery branch at NCI 10 years ago described a T-cell clone derived from a kidney cancer patient that recognized soluble trail presented by DR4 in an MHC unrestricted fashion. So there clearly is much to be known and learned about T-cell recognition and how T-cells with anti-tumor reactivity might ultimately play a role in the T-cell therapy for kidney cancer in the future. And so I've run up my time, but glad this is the last slide. And so I just want to say that the studies that Dr. Sewell's lab had published about using a genome-wide CRISPR screen to identify the antigens recognized by tumor-reactive T-cells has been adapted in our lab, and that's the focus of our DoD, CDMRP, KCRP IDEA Award, and which is illustrated here. I just wanted to end. I could not be more thankful and grateful to the DoD for their support of kidney cancer research. It's an absolute game changer, and we look forward to really presenting the results of these studies in future meetings. So thanks very much. This is the group that does all the work. I just rearrange paper clips all day. But thanks for the opportunity to be here.
Questions?
The case for the 68-year-old woman is astounding, given how many number of years she has. From a resistance standpoint, can you comment on why these tumors are not acquiring alternative drivers? How is single clonal event just evolutionarily rendering these tumors static in some ways? Stop going out.
Yeah, great question. I'd like to think that a reasonably broad and diverse anti-tumor T-cell response is holding that tumor at bay despite the many mutations. So she had Foundation 1 sequencing that revealed at least 18 different pathogenic mutations. And, I think, I'll be honest, my bias, the reason that allogeneic transplantation works for refractory leukemia is that it does enable a polyspecific, polyclonal, broad, deep, and polyfunctional immune response around which escape is impossible. And I think that's what happened in this particular patient.
Is the neoantigenic epitope shared between multiple proteins, somatic mutation by any chance? Is that just very small differences in that epitope still shared?
Across patients or?
So in that patient, across different proteins in that gene.
Yeah, I don't think we know enough about that, but I'd offer that I don't think there's no evidence as yet that the anti-tumor response is against neoantigens as popularly conceived at the moment. Single nucleotide mutations that cause single amino acid mutations in the coding region of canonical genes. I think the point I tried to make is that the peptides presented on the surface of any cell, be it a normal cell or a tumor cell, are a superset of all of the peptides that we might predict from knowledge of the curated human proteome. All kinds of weird peptides are presented on the surface, antisense transcripts, intronic sequences, splice peptides, peptides that are encoded in the untranslated 5' and 3' region of genes. And I think So the target universe is a lot broader than we would otherwise think based on a consideration of, neoantigens per se.
Dr. Borgeles?
So, great talk. I'm here. So I have, so you mentioned different approaches to understand the TCR and what's driving these responses. Obviously, the empirical approach, perhaps even solving a structure of a T cell receptor with a particular, engage with an MHC in a peptide. But then you also mentioned the genomics approach, so using genomics to try to predict, and then you also brought AlphaFold. So my question has to do with how accurate these genomics and AlphaFold approaches are. And since you obviously have the empirical data, so if you match your empirical data with predictions from the genomics with respect to, the TCRs that are being assembled, and how far can you take it with, at predicting what might be that epitope, that peptide, and obviously it's clear from your presentation that it's a lot more complex than most of us thought about. Or the same thing with the AlphaFold. The case that in point that you brought up, it's a situation where you felt, if I understood correctly, that the T cell receptor didn't look like anything you've seen before. So is it an error of AlphaFold, or can we trust it? Could you sort of bring the 3 dimensions together?
Yeah, great question. Thank you for asking. I've spent decades struggling with laboratory approaches to the identification of ligands recognized by T cells. I do think I'm amazed by AlphaFold. I have nothing to do with AlphaFold in general. As you know, over the course of the summer, it predicted the structure of pretty much every known protein in biology. It is by far and away the best protein structure predicting platform out there. I think the sky's the limit. I would not be surprised. Obviously, predicting the antigenic specificity of a heterodimeric alpha-beta T cell receptor is a challenging task, but with enough training data and the right machine learning approaches, I would not be surprised if in 5 years AlphaFold could predict the structure, the antigenic structure recognized by a T cell receptor.
Wayne.
Wonderful talk. I just have a question. So if AlphaFold's predicted that you have 2 protruding loops in the alpha and beta chain, if I understand you correctly, you're suggesting that they may not be recognizing a peptide within MHC. Is that correct?
Yeah, I don't think it does. Yeah.
So, doesn't that really mean that it's a public epitope that you might be able to find by synthesizing that TCR and screening other renal cell carcinoma cell lines to see if you got recognition? It seems like for the advancement in the field that AlphaFold has provided that, it seems that is a sort of a logical experiment that could take you to the next step. I'm just curious your thoughts on it.
Yeah, so armed with the nucleotide sequences of the alpha and beta chains, we can make a recombinant lentivirus and then get that TCR expressed in healthy CD8 or CD4 positive T cells, which is the tool we use to do all of our experiments. And we've done that with that particular Sentinel clone from Judith Ray, and tested it actually against predicted neoantigens from this patient's, given this patient's profile. There was some reactivity with some of the predicted neoantigens. I'll be honest with you, as a T cell immunologist, I don't really think that we've found the true ligand for that particular T cell clone. These studies were described in the paper at the bottom there, but I actually do think, based on the structural evidence suggesting that the CDR1 and 2, which really make contacts with the alpha helices of the MHC molecule, being so really out of the picture, literally, in that particular TCR, that antigenic specificity is exclusively determined by the CDR3 alpha and beta. And given that their length, I think it actually probes a molecule that's probably on the cell surface and not a peptide MHC ligand.
If I may ask a short question, so the frequency of these HLA-independent TCR recognizing other targets, obviously it's intriguing because you can have universal treatment across different HLA types. How often do you think this event occurs? And should we start collecting lymphocytes on everybody who's responding to look for those?
I would say yes, definitely collect the lymphocytes and cryopreserve them. I don't know. I think Dr. Sewell will speak to that.
I'd say it's about 30% of your T cells don't see peptide HLA.
Really?
Overall, numerically.
Yeah.
And I can tell you maybe afterwards what a few of them see. So I'm not talking on this now.
Oh,
because that's another
sorry.
talk, but I don't mind giving it as well, whatever.
Yeah. No, I think it's been underappreciated heretofore, but I do think that a significant fraction of alpha beta T cells do not recognize conventional peptide MHC ligands.
Yeah. Right. So with that perfect timing, I'm going to introduce Dr. Akbay from the powerhouse of UT Southwestern Kidney Cancer Center. She'll talk about overcoming resistance to immunotherapy by targeting telomerase.
Hi, everyone. I want to thank the organizers for giving me the opportunity to be here today. This is my first KCRS meeting, and it's been great. I've learned a lot and really impressed with the community and everybody's friendliness. So I'm going to introduce the concept of targeting telomerase. So we don't have a lot of data in kidney cancer just yet, but I want to get you guys excited about thinking about telomerase and in kidney cancer. So just to recap everyone on this, telomeres are the protective ends of the chromosomes. All the chromosomes have them. Because telomeres cannot be replicated by the DNA polymerases, they get shorter with each cell division. The chromosomes can be lost maybe 25 base pair to 200 base pair with each cell division, so they get shorter. When they reach a certain length, cells can no longer divide. And tumor cells and other cells that need to divide, need to maintain their telomeres. Telomerase is a specialized enzyme that replicates the telomeres. And in most normal adult cells, it's not expressed, so they cannot divide indefinitely. But cancer cells express telomerase. In about 85 to 90% of human tumors, telomerase is expressed. The remaining tumors, they still have to maintain their telomeres. They just adopt an alternative mechanism called ALT, actually named after the mechanism is alternative lengthening, and that's based on a homologous recombination repair. Telomerase function is critical for the cancer cells, and so therefore targeting telomerase could be a universal anti-cancer approach. However, we haven't been able to target telomerase successfully until now. There were previous inhibitors used to target telomerase. This was an oligonucleotide antagonist to the telomerase RNA component, imetelstat, that was previously tested. It's still being tested in the hematological malignancies. It was tested in non-small cell lung cancer with no benefit in PFS or overall survival. For the reason for that is that when you inhibit telomerase, the telomeres don't get shorter immediately. So there is a lag period. So it doesn't work as if you have a platinum-based agent that you can kill the cells immediately. So the imetelstat takes a long time to work, and there's not a lot of time with the patients. A nucleoside analog that was rediscovered by the Shea lab at UT Southwestern it was discovered to be targeting telomerase has been really exciting. So this molecule was tested in leukemia in 1977, and there had some efficacy. However, it wasn't known that it actually has some specific activity with the telomerase or any special connection with the telomerase at the time. There was some activity, but there was, in general, hematological toxicities because the drug was given continuously. Jerry's lab discovered that actually this molecule is a substrate for telomerase and has a higher affinity to telomerase than other DNA polymerases. It gets incorporated into the newly synthesized telomeres and causes uncapping of the telomeres because the proteins that bind and protect the telomeres cannot bind. The telomeres are exposed and they get recognized as DNA damage, or sometimes they fuse with other chromosomes and break, and micronuclei is released to the cytoplasm. So this drug, unlike the imetelstat, causes rapid cell death, and so it can be utilized in a clinic. And cancer cells, we've tested in the lab, and Jerry's lab at UT Southwestern has tested as well, is effective in cancer cells that express telomerase. So we've tested in ccRCC, mouse and human lines, and also small cell lung cancer lines. This is about a 1 micromolar dose for 5 days. We do see the cells are sensitive to this. And to kill a normal cell that does not express telomerase, it would take about 100-fold of this dose. So there is a therapeutic window with this drug with the cancer cells. But what's interesting is that this drug not only kills the cancer cell, but the type of DNA damage that's created with this drug can be communicated to the immune system. So this concept was introduced by another colleague yesterday, that DNA damaging agents in certain cases can cause immunogenic cell death. And so that's through activation of the cGAS-STING pathway. So what was shown by Shea and Fu labs at UT Southwestern is that targeting telomerase caused micronuclei, and in some cases cytoplasmic DNA that was not in micronuclei, and that activated the cGAS-STING signaling in the dendritic cells in the microenvironment, and that activated T cells, and they were effective against killing the immunogenic colon cancer model. It was really effective response against this immunogenic model when mice were implanted with the MC38 model and were treated with only 3 doses of this drug and a low dose. They were cured of their tumors if the mice were immunocompetent, and in immunocompromised mice and NSG mice, this response wasn't seen. There was no response. And mice, when they were rechallenged after about a month with the same tumor, they completely rejected if they'd been previously treated with 6-IODG, while the naive mice were able to grow the tumors. There was systemic anti-tumor immunity and long-term immune memory with this drug. Now, we were interested in testing this in small cell lung cancer, and part of my lab works on small cell lung cancer, and we're interested in that because unlike ccRCC, it's pretty immune resistant and or not as inflamed, actually. Some people refer to it as immune desert. Small cell lung cancer patients do not benefit well from the immunotherapy. It's a very, very, very small percentage of the patients benefit from the treatments. So we are worked on characterizing the microenvironment, and also we're interested in testing this as the agent became available at UT Southwestern through Jerry's group. Just with 3 doses of this drug in this very aggressive metastatic small cell lung cancer immunogenic model, or immune competent model, I should say, it's not immunogenic, we've seen great therapeutic benefit as just with the single agent. And we've also done experiments where we've treated mice where there were tumors were formed after a few weeks of the tumor implantation. And we've also done this with the subcutaneous tissue as well, and we all see a therapeutic benefit. And it's interesting because also liver is known to be immunosuppressed, and that they're usually in lung cancer, having liver metastasis associated with poor response to immunotherapy. So having some activity in the liver mets is very important. So we wanted to understand how this drug might be working, and so we implanted the tumor cells in mice and treated mice with few doses of the drug, and then euthanized the mice to look at the tissue. And we do see on-target drug effects. So we do see that we are inducing the DNA damage, and we are inducing the apoptosis of the cancer cells. But we're also, interestingly, seeing a big change in the microenvironment, that we're seeing influx of immune cells, NK cells, and T cells in the liver. And interestingly, that this work is still ongoing. We've seen an expansion of the TCF1 positive cells that was also in Ari's slides. This population of T cells has been shown to be important for response to immune checkpoint blockade, and the lack of these cells were associated with lack of response. So this is very interesting. This cell population hasn't been reported in small cell lung cancer models. And to take our findings into a more translational and clinically relatable system, so we wanted to use human small cell lung cancer cells. So for that, we've utilized another method that was introduced yesterday, the humanized mouse model system. So in this model, we are reconstituting the immunocompromised mice with the human cord blood cells and giving them about 3 months for them to reconstitute, and then implanting tumor cells and then treating mice with the treatments. Really, without any reconstitution, there is no therapeutic response to 6-DIO-DG in this model. So you really need the immune cells. And there's also some benefit and some synergy with the PD-L1 antibody. And I want to highlight that small cell lung cancer is very immune resistant, and people have even expressed OVA in small cell lung cancer models, and they're still pretty resistant to PD-L1 antibodies. So this is really impressive. Based on the preclinical work done at UT Southwestern, this drug is now being clinically tested in cancers, and now its phase 2 is enrolling in Australia based on the work done in the Shea lab, and there are plans for expanding into colorectal cancer and small cell lung cancer based on the preclinical work. So it's an agent that I hope that will be interesting to you as well. And so we are in kidney cancer meeting. Of course, telomerase activity, as I said, is observed in many cancers. And so this is a paper from about 20 years ago, so it's not the most sensitive method of what we can do now with the telomerase activity, but in ccRCC, telomerase activity is detected in the majority of the tumor cells, but not in the normal adjacent tissue. So that gives us a therapeutic window to treat the cancer cells without damaging the normal tissue. And this is another method of quantifying telomerase activity, and in that too, that we do see there's a difference between the normal adjacent tissue and the tumor tissue that we should be able to target telomerase in kidney cancer. So now really our DoD grant and that what we plan to do is just summarize here. I'll keep it short. We're trying to understand how this drug might be working in kidney cancer and reduce single agent activity in the RENCA model. And we realize there are limitations to this model, and we're interested in testing other models as introduced by Ari, and also potential clinical models through the UT Southwestern Kidney Sport. I'll thank the people who've done the work in my lab and our collaborator, Jerry, who's really is a tremendous help in getting this, and Kidney Sport, and also the DoD funding for funding this project. Thank you.
Open up for some questions now.
That's very interesting work. So my question is, the ccRCC actually is not a telomerase activity inhibitor. It's just decap the telomere protection mechanism. So what's the side effects of this drug in general? And the question is, in the mice, telomere is very long, so you won't see too much side effects. But in the human, it's much shorter than the mouse. So how are you going to make sure it is not that positive, especially for neural cells?
Yeah, that's a great question. So to really get around the problem of that, this is not a telomerase inhibitor, so it really is a nucleoside analog, just has a more affinity to telomerase. To reduce the potential toxicity, we're using really intermittent low dose, and so really a few doses is therapeutically effective. There were also studies done previously in immunocompromised mice, continuous dosing, and mice can tolerate up to 2 weeks of continuous daily dosing of drug, even up to 5 mg per kg. We've tried 2 and 3, and it's been tolerated with just a few doses. So because even the lymphocytes express telomerase activated T cells. So that's why we are trying to schedule the PD-L1 antibodies maybe a few days after this inhibitor is given. So yeah, there is that risk, but I think with the intermittent dosing, we get around that problem.
Esra, so nice talk. What do we know mechanistically about how inhibition of telomerase and the formation of these micronuclei are inducing an immune response? Is it mediated by the STING pathway? What's known? In different models, it seems to be different. With the MC38, they were very much
dependent on the myeloid cell cGAS-STING pathway, as neither the tumor cGAS or the STING was necessary for that response. It's a little bit different in small cell lung cancer, and we're still trying to figure out. So both tumor and myeloid cGAS-STING may be important for the response.
May I ask a question about how do you assess, is there a practical way of assessing the dependence of tumor samples, for example, on telomerase, and would you, a potential biomarker, for example, if you want to build stuff?
That's a great question. I think that's important, and actually like learning from the previous experience with the imetelstat, even though there wasn't overall a significant PFS benefit, the patients that benefited were the ones with the shorter telomeres, for example. That was something because that one takes a long time. There's an assay to determine telomerase activity. So there's one called the TRAP-based assay. That's like a PCR-based assay utilizing lysates from the tumor tissue. And that may require a little bit more sample than there's a digital drop-based assay to determining telomerase activity. So it's possible to do that.
Any other questions? Otherwise, we'll move on.
Yeah. So the next speaker is actually an invited speaker. Susan Poteet have been